there! Now that we've looked at the ways materials move in and out of cells in the previous clip, let's look at some of the factors that affect these processes, such as cell size, characteristics of the materials, and the extracellular environment. When an object is small, it has a large surface area compared to its volume. Let's compare nets of these two cubes. The first cube has a surface area that is 2 centimeters in length by 2 centimeters in height with 6 sides, which is equal to 24 centimeters squared. It has a volume of 2 centimeters length, 2 centimeters height, and 2 centimeters depth, which is equal to 8 centimeters cubed. This gives us a surface area to volume ratio of 24 to 8. That's simplified to 3 to 1. Let's compare that to a bigger cube. The second cube has a surface area that is 4 centimeters in length by 4 centimeters in height by 6 sides, which is equal to 96 centimeters squared. It has a volume of 4 centimeters in length, 4 centimeters in height, and 4 centimeters in depth, which is 64 centimeters cubed. This gives us a surface area to volume ratio of 96 to 64, simplified to 3 to 2. As an object becomes larger, the surface area compared to its volume is smaller. This limits the suitability of diffusion to transport materials in and out of cells. This means that there is a physical limit of the size of a cell due to the efficiency of diffusion. Now let's look at the types of materials. Each mode of transport moves specific materials in and out of the cell. Diffusion moves substances such as oxygen and carbon dioxide. Osmosis moves water and dissolved ions such as potassium and chlorine. Facilitated diffusion by the protein channel in the cell membrane moves glucose and ions. Active transport moves ions, whereas the endocytosis and exocytosis move large molecules, macromolecules, such as bacteria, proteins, and droplets of liquid. The extracellular environment has an important role in the transport of materials in and out of cells. In the process of osmosis, transport of water, it is crucial. There are 3 types of osmotic solutions. Isotonic, where the concentration of ion dissolved in the solutes, the substances, are the same on both inside and outside of the cell. Hypertonic is where the concentration of ions is higher on the outside of the cell than it is on the inside. And hypotonic is where the concentration of the solute is higher on the inside of the cell rather than the outside. These solutions affect the process of getting balance of water and solutes across the membrane. As osmosis happens, more water molecules either enter or leave the cell. When the cell fills, the cytoplasm exerts a greater pressure against the membrane. This stops the production of excess water from entering the cell. This is referred to as osmotic pressure, and it stops the cell from accumulating excess water. However, different cell types can tolerate different osmotic solutions. Both plant and animal cells tolerate an isotonic equal solution really well. Where plant cells tolerate a hypertonic solution high on the outside, because of their large vacuole and sturdy cell wall, whereas an animal cell will rupture. Both plant and animal cells become flaccid or soft in a hypertonic, high on the inside environment, as they move large amounts of water to the outside to balance. There are many factors that influence the transport of materials in and out of cells. See you next time.